Welcome back to the Deep Dive, the show where we take these incredibly complex blueprints of the human body and, well, we try to turn them into something you can actually wrap your head around.
And today we are tackling a big one, a monumental one, really. We're mapping the entire communication network that lets you consciously feel the world and then decide to act on it.
We're going deep into the somatic nervous system, the SNS.
That's right. Our mission today is to follow a single signal from the very beginning. Let's say a gust of cold air hits your skin all the way to the end point, the conscious decision to go and put on a jacket.
And we've got a fantastic set of sources to guide us. They detail every single microscopic step from these tiny little relays in your spinal cord to the massive processing hubs in your brain.
So if you've ever felt a bit overwhelmed by just how many pathways are involved in something as simple as moving a finger or, say, recognizing a song, we are here to clear that up.
Okay, so the somatic nervous system, where do we even start? What's the big picture here?
The first thing to get straight is its functional role. I mean, anatomically, you could say it's part of the peripheral nervous system, but that doesn't really tell you what it does.
Right.
Functionally, the SNS is defined by what it controls: conscious perception of your environment and voluntary control over your skeletal muscles.
That distinction is so important. It's the stuff you are, for the most part, intentionally directing. It's not the autonomic system that's, quietly managing your heart rate in the background.
Exactly. And the basic architecture, when you boil it down, is beautifully simple. It's always sensory input.
The world making contact with your body.
Right. Then central processing.
The central nervous system, your brain, and spinal cord trying to figure out what that input means.
And finally, motor output. The skeletal muscles responding to the command. Input, process, output.
To make that loop really concrete, let's use that classic example, the withdrawal reflex. I think everyone can picture this. You accidentally touch a hot stove.
The sensory input is instantaneous. You have receptors in your skin that scream damaging heat. That signal, it travels at incredible speed along a sensory nerve fiber right into the dorsal root of your spinal cord.
And the dorsal root is like the backdoor entrance into the central nervous system.
It is. Now, the signal hits the spinal cord, and here's the amazing part. Even before your brain is consciously aware of the pain, the spinal cord acts as its own little processing unit.
A local command center.
A local command center. That sensory neuron immediately activates a motor neuron in the ventral horn. That's the forward part of the spinal cord's gray matter where these motor neuron bodies live.
And that motor neuron fires, sending a command right back out.
Right back out the ventral root, straight to your arm muscles, specifically the biceps, causing it to contract and yank your hand away from the stove.
So it's a direct arc. Input, a super fast process in the spine, and immediate output. It bypasses the brain, at least initially, for pure speed.
And that entire sequence is the somatic loop in a nutshell. We're going to unpack all the little details, like how it makes sure the opposing muscles relax so you can move even faster.
Okay, so that's our roadmap. We're following that signal. Section 1, how we translate the world with our sensory receptors. Sections 2, 3, and 4, how the CNS processes all that information from a simple touch to complex vision. And finally, sections 5 and 6, how the brain plans a movement and sends the command to make it happen. All right, let's start where all our knowledge of the world begins, right at that interface between the environment and your body. And our sources point out two terms that are very, very easy to confuse. This is a big exam trap alert: sensation versus perception.
Yes, and drawing a hard line between these two is crucial. Sensation is just the biological event. It's the raw data.
So it's just the receptor cell getting activated by a stimulus.
Exactly.
Yeah.
Think of it as your body's sensor array registering a shift in pressure, a bit of heat, a certain wavelength of light. It's not information yet, it's just data.
And perception is what happens next.
Perception is the interpretation. This happens centrally in the brain. It's the process of taking all that raw data, integrating it, and making it into a meaningful pattern.
So the light hitting my retina is the sensation. But the conscious realization that that pattern of light is a car speeding towards me, that's perception.
You've got it. And that highlights something important. Not all sensations lead to conscious perception. Your internal organs are constantly sensing things. We call them interoceptors, monitoring chemical levels, pressure.
But I'm not consciously aware of my blood pressure second to second.
Not unless it crosses a certain threshold and, causes pain or discomfort. The SNS is mostly concerned with the sensations that do bubble up to conscious awareness and require some kind of action.
Okay, so the outside world speaks in what? Pressure and chemicals and light.
Yeah.
And our nervous system speaks in electrical signals. There has to be a translator.
There does. And the technical term for that translation process is transduction.
Trans.
It's the universal process where energy from a stimulus, be it mechanical force, a chemical, heat, light, whatever, is converted into an electrochemical signal the nervous system can understand.
So how does that work?
Usually, the stimulus physically or chemically changes the cell membrane of a sensory neuron. It might open or close little gates, what we call ion channels. This creates a small electrical change, and we call that initial spark a receptor potential.
It's the fundamental language bridge between the world and our mind. Okay, let's classify the translators themselves, the sensory receptors. We can group them by their structure, their function, and their location. Let's start with what they look like.
Structurally, you can think of it as a spectrum of complexity. On the simplest end, you have free nerve endings.
Sounds basic.
It is. These are just the bare dendrites of a sensory neuron, just sort of embedded in your skin or other tissues. They're not fancy, so they tend to handle general information like pain, temperature, and just crude touch.
A bit raw, but it gets the job done.
Then you move up to encapsulated endings. Here, the nerve endings are wrapped up in layers of connective tissue.
And that wrapping isn't just for protection, is it?
Not at all. It fundamentally changes how the receptor responds. The capsule acts like a mechanical filter. The classic example is the lamellated corpuscle or Pacinian corpuscle. It looks like a tiny onion.
An onion?
Yeah, and because of those layers, a steady pressure won't do much. You need a rapid vibrating pressure to distort those layers and finally activate the nerve ending in the middle.
So the capsule basically tunes the receptor. It makes it super sensitive to one specific thing, like high-frequency vibration, and ignore everything else.
Precisely. It lets you distinguish different textures. And the third type is the most complex, the specialized receptor cell.
Okay.
These are totally separate, distinct cells that are hyper-specialized for one job. Think of the photoreceptors in your eye or the hair cells in your ear. They aren't neurons themselves. They detect the stimulus and then release a neurotransmitter onto a neuron they're connected to.
That covers the structure. Now for function, what kind of energy are they actually translating?
Functionally, they fall into a few big categories. First, chemoreceptors. They respond to chemicals, so that's taste and smell, but also internal things like osmoreceptors checking your blood's saltiness.
And pain, right? Isn't that chemical too?
A lot of it is. Special chemoreceptors, called nociceptors, respond to the chemicals that are released when your tissues are damaged. Pain is literally a chemical warning signal.
Okay, what's next?
Mechanoreceptors. This is the biggest group in the SNS. They respond to any kind of physical distortion, touch, pressure, vibration, sound, the stretch in your muscles.
It's amazing that hearing is basically a very refined sense of touch.
It is. At its core, it's a physical force opening an ion channel. Then you have thermoreceptors, pretty straightforward. They detect changes in temperature. And finally, photoreceptors, the most specific of all. They only respond to the energy of visible light.
Okay, structure, function.
Right.
What's the last way to classify them?
Location, as in where is the information coming from?
Makes sense.
If it's from the external environment, we call it an exteroceptor. These are in your skin, eyes, ears, and so on.
And if it's from inside?
That's an interoceptor. They monitor your internal organs, blood pressure, how stretched your stomach is. You're mostly unaware of these.
And there's a third one. You mentioned it earlier.
Ah, yes. The proprioceptor, an SNS all-star. These are found in your muscles, tendons, and joints. They are constantly reporting on the position of your body in space, how tense your muscles are, how bent your joints are.
This gives us our sense of proprioception, right? My ability to touch my nose with my eyes closed.
Exactly. And also kinesthesia, the sense of body movement. Without proprioceptors, walking would be an incredibly difficult, conscious task. You'd have to watch your feet with every single step.
So we often group all these into two big buckets, general senses and special senses.
Right. The general senses are the ones distributed all over your body: touch, pain, temperature, proprioception. We lump those together as somatosensation.
And the special senses are the ones that have their own dedicated complex organs, all located in your head. Sight, hearing, balance, taste, and smell.
And we'll dive into their special pathways in a bit.
All right, let's pause for a memory hook for section 1. The sensory system converts the world into electrochemical language through a process called transduction, using receptors that are tuned by their structure, their function, and their location. Okay, so we've successfully translated a physical stimulus into an electrical signal. Now, the big question: how do we get that signal up to the brain for conscious perception? We'll start with somatosensation, that group of general senses like pressure, vibration, and pain.
We're now on the spinal highway, and this highway has very, very specific traffic rules. The main rule is that all the major somatosensory paths use a 3-neuron chain.
A 3-neuron chain. Why 3?
It's a way to ensure the signal is processed and relayed properly before it hits the conscious cortex. It's not just a straight wire.
Okay, walk us through that chain. Who's the first neuron?
The first-order neuron is the sensory neuron itself. Its dendrites are the receptor out in your skin or muscle, and its axon travels all the way into the central nervous system, so into the spinal cord or brainstem. Its job is just to collect the raw data.
And the second one?
The second-order neuron lives inside the CNS. Its job is to take that signal and carry it up towards the head. And, crucially, this is almost always the neuron that decussates.
Decussates.
Exactly.
That means it crosses the midline, right? From the right side of the body to the left side of the brain.
Exactly. And it travels up until it synapses in a critical structure called the thalamus.
The famous sensory relay station.
The gatekeeper. And that's where the third-order neuron starts. It picks up the signal from the thalamus and makes the final journey, projecting directly to the primary sensory cortex in the parietal lobe. And it's there, and only there, that the raw data finally becomes a conscious perception.
So you said the second neuron is usually the one that crosses over, and the difference between the two main spinal pathways is where that crossover happens.
That is the single most important functional difference. It's the key to diagnosing spinal cord injuries because it tells you whether the sensory loss will be on the same side as the injury ipsilateral or the opposite side contralateral.
Okay, let's take the first highway. The dorsal column system, or
Express lane. It carries information you need to be very precise about. Fine touch, vibration, and conscious proprioception.
the stuff you need to know exactly where it is and what it feels like read and braille.
Precisely. Now here's how it works. The first-order neuron enters the dorsal root of the spinal cord, and instead of synapsing, it just turns and travels straight up the spinal cord in the white matter of the dorsal columns.
So it doesn't even talk to a second neuron yet. It just goes right up.
It holds onto that raw signal all the way up to the brainstem, and our sources note that it's all very organized. The axons are arranged topographically. Input from your lower body travels in a bundle called the fasciculus gracilis, and input from your upper body travels next to it in the fasciculus cuneatus. It's like a little map of your body ascending the spinal cord.
Okay, so here's the big question. Where does it finally cross over?
And here's the exam trap alert. The DCS crosses very late. The first-order neurons finally end deep in the brainstem, in the medulla, where they synapse in specific nuclei.
Okay.
It's there that the second-order neurons begin. They immediately cross the midline and then ascend to the thalamus in a big fiber bundle called the medial lemniscus.
So fine touch and proprioception from my right hand travel all the way up the right side of my spinal cord, deep into my brainstem, and then cross over to the left side.
You've got it. Now let's contrast that with the other major highway, the spinothalamic tract.
And this one is the warning system, right?
This is the warning system. It's the dedicated path for pain and temperature sensation, the stuff you need to react to quickly.
Okay, so the first neuron enters the dorsal root, just like before.
But then the journey changes immediately. This pathway sacrifices some positional accuracy for pure speed. The first-order neuron synapses with the second-order neuron right away, there in the dorsal horn of the spinal cord.
And the crossover.
The second-order neuron immediately decussates in the spinal cord, usually at the same level it entered, or maybe 1 or 2 levels up. Then it ascends all the way to the thalamus on the opposite side.
Whoa, okay. That is a huge functional difference. Let's say I have an injury that damages just the right half of my spinal cord.
A condition called Brown-Séquard syndrome?
So for the area below the injury, what would I feel or not feel?
This is why knowing the crossing point is everything. Because the spinothalamic tract for pain and temperature crossed immediately, damage to the right side of the cord means you would lose pain and temperature sensation on the left side of your body.
Contralateral loss.
But because the dorsal column system for fine touch and proprioception was still traveling up on the same side, damage to the right cord means you would lose fine touch and position sense on the right side of your body.
Ipsilateral loss. Wow, okay, that distinction is clinically massive.
It's fundamental.
What about sensory information from the face? That's not coming from the spinal cord.
Right. That's handled by the trigeminal system, mostly through the 5th cranial nerve. And what's amazing is that it perfectly mirrors the spinal cord's organization.
How so?
The trigeminal nerve fibers enter the brainstem and then they split. The fibers carrying pain and temperature go to one nucleus, the spinal trigeminal nucleus, which behaves just like the spinothalamic tract. The fibers for fine touch and proprioception go to another nucleus, the chief sensory nucleus, which acts like the dorsal column system.
And do they cross over?
They do. The second-order neurons from both of those nuclei decussate and then travel up to the thalamus together in the trigeminothalamic tract. It's just a beautifully consistent design. The function dictates the anatomy.
All right, that's a lot to take in. Let's do a memory hook for section 2. DCS equals dorsal. It runs up the back. And it has a delayed decussation, or crossing, deep in the brainstem. The spinothalamic tract crosses soon, right in the spinal cord. Okay, we've moved general sensation from the body up to the central processors. Now we're shifting gears to the highly complex, dedicated systems, the special senses.
And our sources immediately flag a huge exception to the rule we just established. We just said the thalamus is the required relay station for all sensory info.
But it's not.
It's not. There's one sensory rebel, olfaction, the sense of smell.
Smell bypasses the thalamus completely.
Completely. The olfactory receptor neurons are up high in your nasal cavity, in the olfactory epithelium. When an airborne molecule, an odorant, dissolves in the mucus up there, it binds to a receptor on these neurons.
And the axons from those neurons have to get to the brain somehow.
They travel through tiny holes in the bone at the roof of your nose, the cribriform plate, and synapse directly in the olfactory bulb, which sits right on top.
So no thalamus. Where does the signal go from the bulb?
It projects directly to the primary olfactory cortex in the temporal lobe. But here's what's truly unique. It also has a massive, direct, unfiltered projection to the limbic system.
The emotional brain, the amygdala, the hippocampus.
Exactly. And that direct connection is why smell is so powerfully tied to emotion and memory. It's why a certain scent can just instantly, vividly transport you back to a childhood memory, totally unfiltered by any higher level processing.
It's an ancient primal sense.
It is. Survival was prioritized over detailed analysis. One other interesting fact. These olfactory neurons are one of the very few types of neurons in your central nervous system that can actually regenerate. They're constantly being replaced.
Okay, let's move to the other chemical sense, gustation or taste. This one does use the thalamus, right?
It does. And we have those 5 main tastes: sweet, salty, sour, bitter, and umami.
The mechanisms for detecting them are pretty different, aren't they?
Wildly different. Salty and sour are simple. For salty, it's just sodium ions Na+ flowing directly through channels into the taste receptor cells, causing a depolarization. For sour, it's hydrogen ions, H+, from acids doing the same thing.
Direct ion flow. Simple, but sweet, bitter, and umami are more complicated.
Much more. They rely on G protein-coupled receptors, a much more complex signaling system. Sweet receptors detect sugars. Umami, that savory taste, is activated by the amino acid L-glutamate, which you find in things like aged cheese or mushrooms.
And bitter seems like the most important one from a survival standpoint.
Absolutely. We have a huge family of bitter receptors because so many natural poisons and toxins are alkaloids, which taste bitter. The system is designed for maximum sensitivity to trigger a defensive gag reflex. It's a do not eat this signal.
So once the taste is transduced, where does the signal go?
3 different cranial nerves carry the signal to the brainstem, where they all converge on the solitary nucleus. From there, it gets relayed to the ventral posterior nucleus of the thalamus, and finally it lands in the gustatory cortex.
Okay, let's switch from chemicals to mechanics. Let's talk about audition hearing.
The inner ear is just an incredible piece of biological engineering. So, sound waves hit your eardrum, the tympanic membrane, making it vibrate.
And those vibrations are then amplified.
They're amplified by that tiny chain of bones, the ossicles malleus, incisius, and stapes. The stapes then pushes on the oval window, which creates fluid waves inside the snail-shaped cochlea.
So we've turned airwaves into fluid waves.
Exactly. And those fluid waves cause a structure inside called the basilar membrane to move. The actual receptor, the organ of Corti, sits right on top of this membrane.
And that's where the hair cells are.
That's where the hair cells are. They have these little bristles on top called stereocilia, which are embedded in an overlying membrane, the tectorial membrane. As the basilar membrane bounces up and down, it creates a shearing force that physically bends the stereocilia.
And that physical bending is the moment of transduction.
That's it. Bending the stereocilia pulls open tiny ion channels. Potassium ions rush in, the hair cell depolarizes, and it releases a neurotransmitter. Mechanical force is now an electrochemical signal.
What's so brilliant about the cochlea is how it sorts sounds by pitch, by frequency.
It's a built-in frequency analyzer. The basilar membrane is physically different along its length. It's narrow and stiff at the base near the oval window, and it's wide and floppy at the apex.
Like a piano strings.
A perfect analogy. High-frequency sounds only have enough energy to move the stiff base. Low-frequency sounds travel all the way to the flexible end. So where on the membrane is vibrating tells the brain the pitch of the sound.
And where does that pitch-sorted signal travel?
From the auditory neurons in the spiral ganglia, the signal goes to the cochlear nuclei in the medulla. Then it takes a complex route, making a key stop at the inferior colliculus in the midbrain, that's important for auditory reflexes, before hitting the thalamic relay, which for hearing is the medial geniculate nucleus. From there, it goes to the auditory cortex.
And how do we locate a sound in space?
That happens very early, down in the brainstem. The brain calculates the interaural time difference, the tiny microsecond delay in the sound reaching your far ear, and the interaural intensity difference, the fact that your head casts a sound shadow, making it a bit quieter for the far ear. It fuses those two clues to create a 3D sound map.
Amazing. Okay, let's cover the inner ear's other critical job, equilibrium, our sense of balance.
Another form of mechanoreception. We have two systems here, one for linear motion, like accelerating in a car, and one for rotational motion, like spinning.
Let's do linear first.
That's the job of the utricle and saccule. Inside them, you have these little crystals, calcium carbonate crystals called otoliths.
Ear stones.
Literally, they sit on top of a gelatinous membrane. When you tilt your head or accelerate, gravity and inertia pull on these heavy little stones, which slides the membrane and bends the stereocilia of the hair cells underneath. It's like a tiny biological accelerometer.
And for rotation, spinning around.
For that, you use the 3 semicircular canals, which are oriented in the 3 different planes of space, like X, Y, and Z axes. When you turn your head, the fluid inside the canals lags behind a little bit due to inertia.
Right.
That lagging fluid pushes on a little gelatinous sail called the cupula, bending the hair cells within it and signaling rotation in that specific plane.
Where do these balance signals go? A lot of it goes straight to the cerebellum, doesn't it?
It does. A huge portion of the signal from the vestibular ganglion goes to the vestibular nuclei in the medulla, and then straight to the cerebellum, bypassing the thalamus. The cerebellum needs that rapid subconscious data to instantly adjust your posture and coordinate your movements.
And this system is also responsible for that incredible reflex, the vestibulo-ocular reflex, or VOR.
Yes, the vestibular nuclei are wired directly to the cranial nerves that control your eye muscles. So when your head moves one way, the VOR instantly commands your eyes to move the opposite way.
Which keeps the image of the world stable on your retina. It's why you can read a sign while walking without the words becoming a blur.
It's an incredible high-speed stabilization program running constantly in the background.
Okay, another memory hook for Section 3. All the special senses, taste, hearing, balance, and sight, are relayed through the thalamus, specifically those geniculate nuclei, with one major exception: the sensory rebel, olfaction, or smell. We've arrived at the final and arguably most complex of the special senses: vision. It provides so much data that a huge portion of our brain is dedicated just to processing it.
It's an immense task. And it all begins with transduction in the retina, that layer of neural tissue at the very back of the eye.
And the retina contains the 2 famous photoreceptor cells, rods and cones.
That's right. Rods are built for low-light vision. They're incredibly sensitive. A single photon can activate one, but they don't see color. They give us our grayscale night vision.
And cones are for color and detail.
Exactly. Cones need much brighter light to work, and we have 3 different types, each one tuned to a different range of wavelengths. One for short waves, which we perceive as blue, one for medium or green, and one for long or red. Your brain creates the entire spectrum of color by interpreting the ratio of activation between these 3 cone types.
Okay, we need to slow down here and talk about visual transduction because it's completely backward from how you think it works.
It is deeply counterintuitive. It's a reverse signaling system. Here's the key. In complete darkness, your photoreceptor cells are actually active. They are constantly releasing an inhibitory neurotransmitter.
So in the dark, they're basically telling the next cell in the chain to shut up.
Precisely. Now, when a photon of light comes in and hits the pigment molecule, retinol, inside the photoreceptor, it causes the retinal molecule to change its shape.
A process called photosomerization.
Right. And that shape change kicks off a chemical cascade that ultimately causes the photoreceptor to stop releasing its inhibitory neurotransmitter.
So light doesn't turn the cell on. Light turns the cell off.
Exactly, and it's that sudden lack of inhibition that signals to the next cell in line, the bipolar cell, that light has been detected. The bipolar cell is finally free to send its own signal forward.
That is wild. Okay, let's talk about visual sharpness, or acuity. Why is our vision so much clearer in the very center?
That's because of a tiny little pit in the center of the retina called the fovea. The fovea is packed almost exclusively with cones, and critically, it has a nearly 1-to-1 wiring ratio.
Meaning one cone photoreceptor connects to one output neuron.
Right, one cone to one retinal ganglion cell, or RGC. In the periphery of your retina, you might have 50 or 100 rods all converging on a single RGC. That makes the periphery very sensitive to dim light, but it makes the image very blurry, very low resolution. The fovea is high res.
And the axons of all those RGCs bundle together to become the optic nerve, right?
They do. And the spot where they all exit the back of the eye is called the optic disc. There are no photoreceptors there at all, which creates our physiological blind spot.
Okay, so the signal is on the optic nerve. Now it has to get processed, which means the information from the right and left visual fields needs to go to the opposite sides of the brain.
And that crossover happens at the optic chiasm. To get the left visual field to the right brain, the axons coming from the medial half of each retina, the half closer to your nose, have to cross over. The axons from the lateral or temporal half of each retina stay on the same side.
So after that chiasm, the right optic tract is carrying the entire left visual field, and the left optic tract is carrying the entire right visual field.
You've got it. And from the optic tract, the signal goes to its thalamic relay, which for vision is the lateral geniculate nucleus, or LGN. The LGN does some initial processing and then sends the signal on to its final destination, the primary visual cortex in the occipital lobe at the very back of your brain.
But the processing doesn't stop there. Once it hits the visual cortex, the signal famously splits into two different processing streams.
It does. First, you have the ventral stream. This pathway runs from the occipital lobe forward into the temporal lobe. This is the, what is it? Stream?
Identification.
Right. Object identification, color recognition, attaching meaning and memory to what you're seeing. Damage to the stream can cause very specific problems like prosopagnosia, the inability to recognize familiar faces.
Now the second stream.
That's the dorsal stream. It runs from the occipital lobe up into the parietal lobe. This is the where is it stream.
Spatial processing.
Exactly. It's responsible for figuring out where objects are in space, detecting motion, and crucially, guiding your body's interaction with those objects like reaching out to grab a cup.
Before we leave the sensory world and move to motor commands, we have to talk about how all this sensory data is organized in the cortex. This brings us to the sensory homunculus.
The homunculus, which is just Latin for little man, is this wonderfully distorted map of your body's surface that's laid out across the postcentral gyrus, your primary sensory cortex.
And the map is distorted for a very specific reason.
A very specific reason. The amount of cortical real estate given to a body part has nothing to do with how big that body part actually is. It has everything to do with the density of its sensory receptors, how sensitive it is.
So that's why on the map, the hands and the lips are enormous.
Enormous. They are wildly exaggerated because they are packed with receptors for fine, discriminative touch. Meanwhile, your entire back, which is physically huge but not very sensitive, gets a tiny little strip of cortex. The map reflects function, not form.
Got it. Okay, a quick memory hook for section 4. The homunculus is distorted because cortical space is allotted based on sensitivity, not size. And vision is processed into two streams. What? Ventral stream to the temporal lobe. And where? Dorsal stream to the parietal lobe. All right, we've done it. We've taken a stimulus from the outside world and turned it into a conscious perception in the brain. Now we have to act on it. The system now shifts from sensory input to motor output, and that whole process begins in the frontal lobe.
It's a hierarchy of command. And the absolute highest level is the prefrontal cortex, right at the very front of your brain. This area is responsible for what we call executive functions.
So this is the big boss, the CEO.
The CEO. It's responsible for planning, judgment, working memory, and deciding on a goal. It answers the question, why should I move? Not how, but why.
And we know so much about this area because of that incredible, famous case from the 1800s, Phineas Gage.
The railroad foreman who had a tamping iron shoot straight through his head, destroying much of his left prefrontal cortex. He survived, and his motor skills and memory were intact, but his personality completely changed.
He lost his ability to plan, to control his impulses.
He became erratic and unreliable. His case was one of the first pieces of evidence that complex intentionality, our personality, resides right there in the prefrontal cortex.
So once the why is decided, the command moves down the hierarchy.
It moves to the secondary motor cortices. Their job is to answer the next question. How am I going to execute this movement? They're the senior managers who draw up the blueprint.
And there are a couple of key players here, right?
There are. You have the premotor cortex, which helps select movements based on external cues, like reaching for a cup you see. It also controls your core muscles to give you the posture you need to support the main movement.
And the other one?
The supplemental motor area, or SMA, this one is more involved in planning movements that are generated internally, especially complex sequential movements you've learned, like mentally preparing the sequence of moves to tie your shoe or play a chord on a guitar.
Okay, so the plan is drawn up. Where does it go for final execution?
It goes to the primary motor cortex, located right in front of the sensory cortex in the precentral gyrus. This is the final output station. It's the go button. It houses the giant upper motor neurons that will send the command down to the body.
And just like the sensory cortex, the motor cortex has its own distorted map, the motor homunculus.
It does. But this time, the exaggeration, the distortion, is based on a different principle. It's not about sensory density. It's about the degree of fine motor control required.
So areas that need lots of precise, dexterous movements get more brain space.
Exactly. Which means, once again, the fingers, especially the thumb and the lower face, for the incredibly complex movements of speech, are gigantic on the motor map. They require a huge number of tiny motor units for that fine control. Meanwhile, powerful but simple muscles, like your glutes, get very little space.
The hands and face dominate both maps, just for different reasons. Okay, the command is ready to go. Let's send it down the descending pathways.
These main voluntary pathways are called the pyramidal system because the fibers form pyramid-shaped bundles in the medulla. They start with the upper motor neurons in the primary motor cortex.
And they split into two main tracks.
First is the cortical bulbar tract. Bulbar refers to the brainstem. This tract controls the motor nuclei of the cranial nerves. So it controls your face, your jaw, your tongue, your eyes. It's largely ipsilateral, controlling the same side of the face.
And the second, much bigger one.
The corticospinal tract. This one controls the rest of the body. It travels down. And then in the medulla, at the very bottom of the brainstem, we have the main event, the pyramidal decussation.
The great crossing over. This is what gives us contralateral control of our bodies.
This is it. About 90% of the fibers cross over to the opposite side here. They form the lateral corticospinal tract, which travels down the lateral part of the spinal cord. This tract is responsible for controlling your appendicular muscles, your limbs. It's all about fine, voluntary control of your arms and legs.
You have the 10% that don't cross.
They continue down on the same side as the anterior corticospinal tract. They mostly control your axial muscles, your trunk and core. This tract provides the stable postural platform you need to be able to move your limbs.
So that's the voluntary conscious command system. But movement is way more complex than that. There's a whole other system working in the background, right?
The extrapyramidal system. This is everything else. All the descending motor pathways that don't pass through those medullary pyramids. They are essential for modulating movement, for muscle tone, posture, balance, all the automatics.
The subconscious support crew. What are the key tracks here?
There are three really important ones to know. First, the vestibulospinal tract. This one gets its input from the vestibular nuclei or balance center, and its job is to constantly adjust your muscle tone to keep you upright and balanced against gravity.
Okay.
Second, the tectospinal tract. This one starts in the superior colliculus, a part of the midbrain that gets visual input. It helps you reflexively turn your head and orient your eyes towards a sudden visual stimulus, like a flash of light.
And
The reticulospinal tract. This is a broad system that originates in the reticular formation of the brainstem, and it heavily influences your overall muscle tone and posture, especially during locomotion.
So the cortex issues a command, but it seems like there's another major player that makes sure the command is actually carried out smoothly and correctly.
And that is the cerebellum. The cerebellum is the ultimate error corrector.
How does it do that?
It's constantly getting two streams of information. It gets a copy of the motor command that the cortex just sent out, the intended movement. And at the exact same time, it's getting a flood of real-time proprioceptive feedback from the muscles and joints, the actual movement.
And it compares the two.
It compares the intention to the execution. If there's a mismatch, Say you intended to take a normal step, but you slipped on some ice. The cerebellum instantly calculates the error.
And sends out a correction.
Instantly. It sends corrective signals, and notably via the cerebrospinal tract, to adjust the movement on the fly, to keep it smooth and coordinated. This is why people with cerebellar damage have such jerky, uncoordinated movements and intention tremors.
So we finally brought the command all the way down. The last step is the lower motor neurons.
The final common path. Their cell bodies live in the ventral horn of the spinal cord's gray matter, and their axons go directly out to the skeletal muscles. They release acetylcholine at the neuromuscular junction, and that makes the muscle contract. It's the final, reliable, do this signal.
And a single one of these lower motor neurons, plus all the muscle fibers it controls, it's called a motor unit.
It is, and the size of that motor unit determines its function. For big power muscles, like your quad, one motor neuron might connect to 1,000 muscle fibers. But for fine precision, like the muscles that move your eye, one motor neuron might only connect to 10 fibers. That's how we get the incredible dexterity in our hands and face.
We started this entire deep dive with the hot stove analogy, so it feels right to end the functional loop by going back to it and really detailing the reflex arc, the simplest and fastest circuit in the whole system.
It's the perfect example. It operates almost entirely at the level of the spinal cord, bypassing the brain for sheer speed. So let's look at that withdrawal reflex again and see the hidden sophistication.
Okay, the sensory neuron detecting the heat enters the dorsal root of the spinal cord.
And it does two critical things at once. First, the main branch of its axon synapses directly with the motor neuron that controls the agonist muscle, the one that's going to pull your hand away, in this case, the biceps.
So it excites the bicep. That's agonist excitation.
Right. But at the exact same time, a little collateral branch of that same sensory axon does something else. It synapses onto a tiny neuron called an interneuron.
An intermediary.
An intermediary. And this interneuron's job is to be inhibitory. It releases an inhibitory neurotransmitter onto the motor neuron that controls the antagonist muscle, the triceps, which would oppose the movement.
So it actively silences the muscle that would fight the withdrawal.
It shuts it down. This is called reciprocal inhibition, and it is absolutely essential for a fast, smooth, unimpeded reflex. You excite the muscle you need and inhibit the one that would get in the way. All in a few milliseconds, all within the spinal cord.
That is just elegant design. And we see that same principle in the stretch reflex, like the knee-jerk test at the doctor's office.
Exactly. The purpose of the stretch reflex is to maintain a constant muscle length. The receptor here is the muscle spindle. When the doctor's hammer taps your patellar tendon, it briefly stretches your quad muscle.
And the muscle spindle fires.
The spindle fires, and its sensory axon goes into the spinal cord and synapses directly onto the motor neuron for that same quad muscle, causing it to contract and kick your leg out. It's a reflex to resist being stretched. And at the same time, an interneuron inhibits the antagonist muscle, the hamstring.
We also have protective reflexes that involve the cranial nerves.
Like the corneal reflex, the blink reflex. If something touches your cornea, the sensory input is carried by the trigeminal nerve. That signal goes to the brainstem, which immediately sends a motor command out via the facial nerve to the muscle that closes your eyelid.
A very important reflex for protecting your eyes. And finally, there's one reflex that neurologists use all the time because it tells them about the health of the entire descending motor system.
The Babinski reflex.
Right. What's happening there?
Well, in an infant, if you stroke the sole of the foot, the big toe will extend upward and the other toes will fan out. This is normal because their main descending motor pathway, the corticospinal tract, isn't fully myelinated yet. It's not mature.
But in an adult, that's a bad sign.
In an adult, the proper response is for the toes to curl downwards. If an adult shows that infant-like response, the toes fanning out, we call that a positive Babinski sign. And it's a huge red flag. It suggests there's damage or a problem somewhere along that primary corticospinal tract, an upper motor neuron lesion. It's a simple bedside test with profound implications.
Okay, final memory hook for Section 6. All of these simple reflexes are circuits that have two critical safety components: agonist excitation to make the move, and antagonist inhibition to make it fast and smooth.
Wow.
What an absolutely exhaustive journey through the somatic nervous system. We have followed the flow from raw energy hitting a receptor all the way to a finely tuned voluntary muscle contraction.
We really have. We established the SNS as the system for conscious perception and voluntary action, all powered by transduction. We saw how general touch is split into two pathways: the high-fidelity dorsal column system, which crosses late in the medulla,
And the urgent spinothalamic tract for pain and temperature, which crosses immediately in the spinal cord.
We then explored the special senses, noting that almost everything, taste, hearing, vision, balance, has to pass through the thalamus as a relay, with the one famous exception being olfaction or smell.
And we learned that the cortex creates a topographical map of the body called the homunculus, where the brain space is allocated not by physical size, but by sensory sensitivity or the need for fine motor control.
And finally, we traced the motor command down from the top, from the executive planning in the prefrontal cortex to the blueprinting in the secondary motor areas to the final go signal from the pyramidal system.
And we highlighted that critical crossing over point. The pyramidal decussation in the amygdala gives us that contralateral control of our limbs.
And we can't forget the background crew. The extrapyramidal tracts that maintain our posture, and the cerebellum, which acts as the ultimate error corrector, constantly comparing our intention with our action to keep us smooth and coordinated.
Okay, I think it's time for a rapid review. Let's lock in some of these key concepts.
Let's do it. What type of structural receptor uses a connective tissue wrapping, like an onion, to make it sensitive to vibration?
That would be the encapsulated endings, like the Pacinian corpuscle.
Which sensory pathway carries conscious proprioception, and where does it decussate or cross over?
That's the dorsal column system, and it crosses late, deep in the brainstem in the medulla.
Okay. In the inner ear, which structure is responsible for detecting linear acceleration using those tiny calcium crystals?
That's the utricle and saccule with their otoliths.
For vision, what's the difference between the dorsal and ventral processing streams?
The ventral stream goes to the temporal lobe and answers, "What is it?" for identification. And the dorsal stream goes to the parietal lobe and answers, "Where is it?"
Yeah.
for spatial location.
Perfect. On the motor homunculus, why are the hands and face so enormous?
Because they require the finest motor control and have the greatest number of small motor units.
Last one. Which descending extrapyramidal tract is critical for using balance signals to adjust your posture and stop you from falling?
That's got to be the vestibulospinal tract.
You got it.
So we've spent this entire deep dive establishing how this incredibly complex system allows us to consciously perceive the world and perform nuanced actions like writing or playing an instrument. But when you step back and look at all the high-speed, autonomous correctional programs built into the system,
The vestibulo-ocular reflex stabilizing your vision, the cerebellum constantly fine-tuning every single movement, the stretch reflexes keeping your posture stable without you even thinking about it.
it leaves you with a really provocative question.
How much of what we experience as a conscious, deliberate, voluntary action is truly an original command from our prefrontal cortex? And how much of it is just our conscious mind experiencing the final, heavily edited and automatically corrected version of a movement that was almost entirely managed by a network of subconscious safety protocols?
We perceive this perfect, seamless intentionality. But maybe that feeling we call control is just the brain's brilliant illusion, its seamless presentation of its own instantaneous autonomous self-correction. Something to think about next time you feel completely in charge of your own movements. Thanks for joining us for this deep dive into the somatic nervous system.